Hey Waxahachie Global, welcome to Chapter 17. This is the last major chapter for this semester. We will cover a small amount of information from Chapter 18, but it won't be a large amount, and I'll try to limit it because our test is coming up very fast. So Chapter 17 is all about basically transcription and translation. We've already talked in Chapter 16 about how we create new copies of DNA and pass on that genetic material. But now we need to know, what does that DNA do? All that instruction code in there contained inside that DNA, it does us no good if it doesn't actually get put to use. So we're going to talk about transcription and translation, which is the process of taking that code, that information that's encoded in DNA, and then using that to create proteins and enzymes, which is the vast majority of what you are made up of, and which is the vast majority of what your body needs to do the various things to keep it alive. So this is From Gene to Protein, Chapter 17. We'll start with a quick overview here. One of the cool things to note is that this whole process that we're going to be discussing in this chapter, called gene expression, basically taking those sections of DNA that we call genes and expressing them, or actually doing something with them so that there's some sort of phenotype that we can see. The process of gene expression is pretty similar in all of the three major domains of life. So whether it's a eukaryote or a prokaryotic creature, or even the domain of archaea, a mushroom, a bacteria, an oak tree, a dog, a whale, all use a similar process to create proteins using the information encoded in DNA. So our overview says the information content of DNA is in the form of specific sequences of nucleotides. We know that DNA is made up of nucleic acid, and there are 4 nucleotides that we have already discussed, and we'll discuss that a little bit more. So the DNA is encoded into these 4 nucleotides. The 4 bases are adenine, guanine, cytosine, and thymine. And we already talked about how adenine pairs with guanine and cytosine pairs with thymine in the DNA chain. The DNA is inherited by an organism from its parents, of course, and that leads to specific traits that we see, the phenotype that we see. And the phenotype that we see, whether it's hair color or shape of nose or number of toes, that is dictated or controlled by the creation, the synthesis of proteins. Proteins are basically the link between genotype and phenotype, between the genetics of a creature and then what you actually end up seeing as an end result. So we have this term called gene expression. Gene expression is the process by which DNA directs certain proteins to be synthesized, created, and it basically includes 2 stages called transcription and translation. So in this chapter, what we're trying to get at is basically an explanation. For example, you have this deer here, this albino deer. That is a phenotype that we see, and it's a major change. I mean, this is a major difference between this albino deer and the normal deer that you would expect to see. So how does his genes or genetics, how does perhaps one faulty gene result in such a dramatic difference, a dramatic change in phenotype? How does one little change in that DNA code or that genetic code end up causing such a large change on the outside that we can see? So that's what we're going to talk about, transcription and translation. We're going to start with concept 17.1, and that says genes specify proteins via transcription and translation. So a gene basically is going to be an instruction code that tells the body to create a specific type of protein, and it will tell it how to put that protein together. So basically think of it as an instruction code like a recipe or like a blueprint to build a house. You have a list of ingredients, and then you have an order in how to put those ingredients together to get your final product. Like we've done in some previous chapters, we want to start with a little bit of background or history. So how was this fundamental relationship between genes and proteins first discovered? Way back in the early 1900s, in 1902, there was a British physician, medical doctor, Archibald Garrod. It might be Garrod, it might be Garrett, I'm not sure. First suggested that genes dictate phenotypes through enzymes that catalyze specific chemical reactions. So Archibald Garrod was one of the first people to say, okay, I think the way that this works is that the genes control the creation or the work of certain enzymes. And we know that enzymes are generally proteins, chemical factors here that promote a chemical reaction. So he said these enzymes control certain chemical reactions that control what we see about the body. So he thought, for example, he was looking at various inherited diseases, and he thought perhaps various inherited diseases were the result of a non-working enzyme. So perhaps you had a disease because there was an enzyme in your body that didn't do what it was supposed to do. And that probably didn't do what it was supposed to do because there was a problem with the instruction code, a problem with the genes. So linking the genes to the creation of an enzyme required an understanding of what we call a metabolic pathway, which we've already discussed in some previous chapters. Basically a series of steps that the cell goes through or the body goes through to produce something or create something or carry out a particular instruction code. So he was the first to say, he was looking at diseases, but he was saying, I think this person has this disease because their body doesn't create a particular enzyme. And their body doesn't create a particular enzyme because the gene that tells the body to do that is faulty or wrong in some way. So Gorad's ideas were really pretty much ahead of his time. I mean, think about 1902 and think about what we've discussed so far in some of our previous chapters. Not a lot of study in genetics was going on at this time. Not a lot yet was known. So his ideas were pretty much ahead of his time. And just like Gregor Mendel's ideas, they weren't really looked at carefully or more carefully understood and studied until sometime in the 1930s. So just like with Mendel, Gorad's ideas were not really picked up on until the 1930s. If you want more information, look on page 326. Gorad was actually studying an interesting genetic condition, and it's called, and I'll have to work to pronounce this, it looks like alkaptonuria, yep, I'm going to go with alkaptonuria. So take a look on page 326. This disease or this condition, alkaptonuria, actually turns your urine black. So imagine having a condition where every time you go to the bathroom, your urine comes out black. Well, that's what he was looking at. So take a look on page 326 for more information there. So let's fast forward through the 1930s to about the 1940s, where some of the next major work or discoveries were made. We're going to be talking about two particular scientists, George Beadle and Edward Tatum, and they were working at Stanford University in the early 1940s, and they were looking at bread mold. Bread mold made for a good genetic test subject because it had a fairly simple genetic code, not a lot of chromosomes. So what they were doing was they were bombarding bread mold with X-rays, and they were trying to purposely create mutations within the genetics, and they knew that radioactivity would do this. So they were exposing bread mold to X-rays, and then they were trying to create mutants of varieties of this bread mold. The mutants they were trying to create would be the type of mold that would not be able to survive on something called minimal media. Minimal media would be like the gel material that you put in the agar, the agar material that you put in the petri dish, that only has some essential nutrients in it. It only supplies some basic nutrients like salt, glucose, and biotin. The normal mold can actually survive just on these minimal nutrients because it can produce itself all the other nutrients that it needs. But they were trying to change the genetics of the mold and make it so that one of the mutants could not survive on just the minimal media. They ended up creating basically three types of mutants, mutant mold that could not survive on the minimal media. And then they tested these mutants in different vials with the minimal media, and then they added one nutrient to see which nutrient that the mutants could not synthesize. So basically they were doing a test where they had their little mold spores on the media, and then they would add a particular nutrient, a different nutrient to each of the petri dishes, and they were looking to see which one is the nutrient that the mold could not produce on its own, that it had to have to survive by being given by the scientists. So what they did, they used these crosses, and they basically came up with three what they called arginine deficient mutants. So these were three varieties of mold that could not create the arginine material that they needed on their own. Each of their three mutants lacked a particular enzyme that was necessary to create or synthesize arginine. Did I say arginine? Arginine? Either way is fine. So they developed what they called a one gene, one enzyme hypothesis, which states that each gene dictates or controls the production of a specific enzyme. And they felt that their hypothesis was accurate because each mutant in their experiment was defective in a single gene. So basically what they came up with was this idea that every single gene controls the creation of one particular enzyme. And so using their mutant mold and their various tests, they developed this idea that held for quite a long time, a one gene, one enzyme hypothesis, where every single gene controls one single production of one particular enzyme. On page 327, there's a nice overview of their experiment with an illustration here. So this is what they did. Basically, they had the minimal media, which is like their control group, and the only mold that could grow in nothing but the minimal media was the wild, the original unaltered mold. And then they had all these different combinations, 1, 2, 3 different mutants, and there were their different conditions. The minimal media with adding ornithine, citrulline, and arginine. And then they were looking to see which ones could live and which ones could not live. And basically what they found was that certain mutants could not live without being given certain nutrients. And if you see, there was basically a pathway that had to be followed for these mutant molds to survive. And what the results really show is a metabolic pathway. So to create all the nutrients to survive, the mold had to go through a number of steps, using gene A and then taking what was made with gene B, taking what was made with gene C, and providing everything that the mold needed. So if the mold was lacking in gene A, well, that's breaking the link at the very beginning of the chain. Now, if you provided something after that, the mold could continue on down the line. If it was missing or had a defective gene B, that was a broken link about in the middle of the chain. So if you gave it what came before, it still couldn't survive because it would stop right there at the break. But if you gave it what came after, then it could continue. And gene C was like a link near the end of the chain. If you gave it what came before that, it wouldn't survive because, once again, it would get to the broken link and be done. But if you gave it what came after, then it would survive. So imagine you have a car that is traveling down to the Florida Keys and it has to cross a series of bridges to get to its final destination down there in the Florida Keys. Well, if you have the first bridge broken, well, then you stop right there and you can go no farther. If you have a bridge broken midway in the path, well, you can go farther, but you're going to stop and not get there. And then if you have a bridge broken at the very end of your travel, well, that's going to get you this far, but you're still not going to get there all the way. So if you could lift the car up and over this broken bridge, then you could continue. And the same thing all the way down the line. So notice that the mutants can only grow if supplied with a compound made after the defective step, because this allows it to bypass that defect and keep on going. So all in all, the basic summary is that their experimentation gave them the hypothesis of one gene for one enzyme. That one gene is basically a code or instruction for one particular enzyme. Well, this hypothesis held for quite a while, but it had to be further developed as we learned more information, because some proteins are not enzymes. For example, the keratin that makes up your nails and your hair and the insulin that your body uses to regulate your sugar, those are not enzymes. So researchers later revised the hypothesis to make it say one gene equals one protein. And that's almost even up to today. That's still kind of how we think about it, one gene, one protein. Many proteins, though, are composed of several polypeptide chains, and each of those polypeptide chains can have their own gene for creating them. So if you really want to be accurate, the Beadle and Tatum hypothesis is probably now best restated as one gene equals one polypeptide, although commonly we still say one gene equals one protein, but more accurately it would be one gene equals one polypeptide chain. So one gene, one protein, you'll hear that pretty often. But again, to be most accurate, you might want to say one gene, one polypeptide. So let's go over just a quick summary of the basics of this process of transcription and translation. We're going to be talking about ribonucleic acid, RNA, very similar to deoxyribonucleic acid. The only difference is the sugar, the ring-shaped sugar on the sugar phosphate base, our backbone, is slightly different. It contains one more oxygen than the DNA one does, and then some of the nucleic acid, the base pairing we'll talk about is slightly different there too. So the first part we're going to talk about is transcription. That is the synthesis of RNA using information in DNA. So we're going to take DNA and use that to make a type of RNA. And the transcription process produces what we call messenger RNA. We're actually going to see 3 versions of RNA, and so we put a little letter beside them to indicate which one we're talking about. And the first one that we're going to look at is called messenger RNA, and you'll see why. So it gets a little mRNA. So we have transcription, which is the creation of messenger RNA. And then the second part is translation. Translation is the synthesis of a polypeptide using information from messenger RNA, and that actually gets done in the ribosomes. So we use DNA to make messenger RNA to make a protein or a polypeptide chain in the ribosomes. Now, like we said, RNA is a single-strand nucleic acid, so that makes it slightly different, where DNA is a double helix, double strand. RNA is a single-strand acid, nucleic acid, made up of a polymer chain of nitrogenous bases. A little bit different than DNA because, number one, it's single-stranded. Its sugar phosphate backbone contains the sugar ribose instead of deoxyribose, and it differs in one nitrogenous base. In RNA, in DNA we have A, C, G, and T, but in RNA we have A, C, G, and U. Uracil replaces thymine. So in RNA there is no thymine. Instead, the nitrogenous base that combines with guanine is uracil. So those are the differences between the two, but they are both nucleic acids. While this transcription translation process pretty much occurs in all living creatures, it's been most studied in bacteria and eukaryotes. So we're going to look at both bacteria and eukaryotes. There are a lot of commonalities between the two, and we'll kind of point out where they do become slightly different though. In prokaryotes such as bacteria, the translation of the messenger RNA can start even before transcription is finished. Remember, they don't have a nucleus, so this whole process can occur sort of all at one time there in the cytoplasm. In eukaryotic cells, it's 2 separate processes because transcription happens within the nucleus. There is a nuclear envelope that separates the nucleus from the rest of the cell, so transcription occurs inside the nucleus, and then that messenger RNA has to go outside of the nucleus to where the ribosomes are to do the whole translation portion. So eukaryotic RNA transcripts have to travel outside of the nucleus before translation occurs, and also eukaryotic RNA usually is modified or changed slightly before it heads out to start the translation process. So you can think of the initial RNA transcript, the initial messenger RNA that gets created, sort of like the instructions from an overworked boss, where he may write out a quick set of instructions on a notepad and throw them out to all of the workers. Well, those instructions, before they go out company-wide in an email, they're going to get cleaned up, they're going to get spell checked, they're going to get formatted by the boss's executive assistant or secretary. He or she's going to type it, proofread it, etc. So that's kind of what's going to happen inside the cell too. The DNA creates this RNA transcript, but it's kind of a messy, quick version of, and then that's going to have to be cleaned up and modified before it gets sent out to the ribosomes to do the actual work. That initial messy set of directions is called the primary transcript. That's the initial RNA transcript from any gene prior to processing, and then we'll find out later how that kind of gets changed and cleaned up a little bit before it gets sent out to the ribosome. We have something called the central dogma, which you perhaps have heard before. This was actually proposed by the DNA double helix co-discoverer Francis Crick in the 50s, 1956. And the central dogma is kind of this idea or concept that explains cellular chain of command. And the central dogma is DNA to RNA to proteins. Now that's simplified, but that is perhaps something you've heard already in your biology classes. The central dogma of the cell is DNA to RNA to proteins. We have a string of nucleic acids, which is used to create another string of nucleic acids, which is then used to create a string of amino acids. So we kind of have one language being translated into a different kind of chemical language, DNA to RNA to protein. There you go. If you didn't get it into your mind enough, there's a picture. DNA to RNA to protein. That's the central dogma, which you often hear in biology class. Let's take a quick look at an illustration for a quick overview, and we'll compare the bacteria cell and the eukaryotic cell. Bacteria cell is over here on the left. It doesn't have a nucleus, so all of this process can occur almost at the same time and kind of in the same location in the cytoplasm area there. So we have DNA. Transcription is taking DNA and making a messenger RNA. So we use transcription process to create messenger RNA, and then the messenger RNA takes that information to the ribosomes, and that's translation, where the ribosome uses the messenger RNA to create a polypeptide chain, which will eventually become some sort of protein. So you notice this occurs kind of all in the same space, and it can even occur basically at the same time. Over here on the eukaryotic cell, which is like you and I, the nucleus is present here. The whole transcription process from DNA to RNA happens inside the nucleus. So we have transcription to what we call pre-RNA or pre-mRNA. That's the messy message from the boss that we're going to get cleaned up. Then we have what we call some RNA processing that we'll discuss in just a moment. That gives you your final messenger RNA. That is transcription. Then it has to leave the nucleus, go out to the cytoplasm, and use the ribosomes to create a polypeptide chain based on that messenger RNA. That's translation. So in a eukaryotic cell, these are two separate processes. They happen at a slightly different time and in a slightly different place. In bacteria cells, like prokaryotic cells, it can occur sort of all at the same time and place. So again, here's the bacteria cell. Here's the transcription portion and the translation portion. In the eukaryotic cell, we have transcription into the first initial pre-messenger RNA. That gets cleaned up, and then that completed messenger RNA leaves the nucleus, goes out to the cytoplasm. The ribosomes use that as a template to create a polypeptide chain, which will eventually make into a protein of some kind. Let's take a quick break right there, and then we'll come back and we'll talk about the genetic code just a little bit, and we will discuss how these instructions are made into proteins, the whole kind of messy biological process there. So let's take a quick break, come right back.